You know, inside every single one of your cells right now, there is literally a biological programming language running the show. It's what makes you you. Welcome to This Explainer. Today, we're diving into one of the absolute coolest, most mind-bending puzzles in the history of science, cracking the genetic code. Because figuring out what DNA looked like was a huge deal, sure, but deciphering how it actually translates into the building blocks of life, now that was the ultimate biological riddle.
So, here's our roadmap for the explainer today. We'll start with a genetic language problem, move into deciphering the code, lay out the rules of the code, look at mutations or genetic typos, and finally wrap up with shifting the reading frame. Okay, section 1, the genetic language problem, translating the code. To understand the problem, we have to look at the two completely different chemical languages happening inside our cells. We've got the language of nucleic acids, and we've got the language of proteins. But trying to translate between them, it creates this massive mathematical mismatch. Think about it. How do you get 20 different amino acids out of an alphabet that only has 4 DNA bases? If one base coded for one amino acid, well, we'd only have 4. If they paired up in 2s, 4 squared is just 16. We're still short. So how on earth does nature bridge this impossible mathematical gap?
Well, the theoretical breakthrough for this biology problem actually came from a physicist, George Gamow. He basically just crunched the numbers and realized, okay, the code has to be read in 3s, a triplet code. If you take 4 bases to the power of 3, it generates 64 possible codons. Now, 64 is way more than the 20 amino acids we strictly need, but mathematically speaking, it was literally the only way life could work.
Section 2, deciphering the code, the scientific Avengers. So Gamow had the math, but actually proving it in a lab, that was a totally different beast, and it took a whole multidisciplinary team. After Gamow proposed that initial 3-letter triplet theory, Hargobind Khurana stepped in and chemically synthesized RNA molecules with highly specific base combinations. Then, Marshall Nierenberg created this ingenious cell-free system for protein synthesis to test it out, and Severo Ochoa discovered the enzyme needed to help polymerize it. Working together in parallel, these scientific Avengers finally mapped out the entire checkerboard of our genetic code.
Section 3, rules of the code, the grammar of life.
Just like English or any spoken language, our DNA has strict grammatical rules we have to follow. First off, as Gamow predicted, it's a triplet code. 61 combinations code for amino acids, while 3 act as biological stop signs. Second, it's read contiguously. That means no commas, no spaces, just continuous 3-letter words read back to back. And third, this code is nearly universal. The exact sequence coding for an amino acid in some microscopic bacterium is the exact same code being used inside your body right this second. Pretty amazing, right? Now, you'll often hear the code described as degenerate. Usually, that's not a great word, but in genetics, it's literally a lifesaver. Because we have 64 functional codons, but only 20 amino acids, it means multiple different codons and actually code for the exact same amino acid. This degeneracy acts like a built-in evolutionary shield. It gives our genetic language a ton of vital redundancy to protect us from accidental errors.
But out of all those triplets, one stands entirely above the rest, AUG. It actually serves a brilliant dual purpose. On one hand, it provides the code for the amino acid methionine, but on the other hand, it acts as the master initiator. Think of AUG as the capital letter at the very beginning of a sentence. It's the exact signal that tells your cellular machinery, hey, start reading the gene right here. Section 4, mutations, the genetic typos.
So what exactly happens when our DNA accidentally gets altered? Well, the whole relationship between genes and DNA becomes super clear when we see what happens when the rules get broken. The simplest error is what's called a point mutation. This is a change in just a single base pair. It's the biological equivalent of a tiny typo, swapping out one single letter in a massive manuscript. Seems pretty harmless, right?
Well, not exactly. Take the sequence for the beta-globin chain. Normally, the DNA sequence perfectly codes for the amino acid glutamate. But if a point mutation happens and swaps just one single letter, suddenly glutamate becomes valine. That one tiny typo literally warps the physical structure of red blood cells, and it results in the devastating systemic human disease we know as sickle cell anemia. Just one wrong letter, with massive, massive consequences.
Section 5, shifting the reading frame. If you thought a simple letter swap was destructive, watch what happens when we accidentally insert a letter.
Let's look at a simple English sentence. Step 1. Ram has red cap. Now, step 2, we insert a rogue letter, B. Because our cells read strictly in triplets without any spaces, it forces the entire reading frame to shift over. So step 3, it now reads, Ram has Bray decap. We haven't just changed a single word, we've completely scrambled the entire rest of the sentence. This is called a frame shift mutation, and it absolutely destroys the resulting protein. But nature actually has a fascinating loophole here. Because the code is read in threes, if you insert or delete exactly 3 letters, an entire codon, the reading frame essentially stays perfectly intact. You're just adding or removing one complete word. The resulting protein might be a little bit longer or a little bit shorter, sure, but the rest of the genetic sentence still reads perfectly from that point on. All right, to wrap things up, I want to leave you with a quick puzzle to test what we've covered in this explainer. Going from RNA to amino acids is pretty straightforward, but what about doing it in reverse? If I handed you a protein sequence, let's say a methionine followed by a string of phenylalanines, could you easily predict the exact RNA nucleotide sequence that built it? Think back to that rule of degeneracy we talked about earlier. With multiple codons for the same amino acid, how many different RNA combinations could potentially spell out this exact same protein? It's a lot, right? Something to think about. Thanks so much for joining me, and keep exploring the code.